Hello bachcho, the question is, in vitro fertilization is a technique that involves transfer of which one of the following into the fallopian tube. So in vitro fertilization or test tube baby program, it is an ART or assisted reproductive technology which helps the infertile couple to have a child. So here the first step of in vitro fertilization. So in vitro means inside the test tube, means here the fertilization is the fusion of gametes. So fertilization is achieved in vitro, it means inside the test tube, in laboratory or in controlled condition. So the first step is collection of semen from the male, it may be the husband or any other donor, or the collection of ovum from the female, which may be the wife or any other donor female. Now they are made to fertilize. Semen is having sperm, so this is the concentrated semen with a huge count of some sperms. So this is made to fertilize the ovum. Now after fertilization, zygote is formed. Now either this zygote or it undergoes cleavage to form the embryo. So embryo up to 8 blastomere stage. So it is transferred into the fallopian tube of the female, which may be the genetic mother or the surrogate mother. So in any case, if the zygote or embryo is to be transferred before 8 blastomere stage, then it is to be transferred into the fallopian tube. And this technique is called the ZIFT or zygote intrafallopian transfer. While if the zygote is undergoing cleavage and it has formed the embryo above 8 blastomere stage. So if it is above the morula stage, if it is above the, means it is in the morula stage or above the morula stage, it means above 8 celled stage it is, then it is transferred into the uterus of the female. Now this technique of transferring the embryo into the uterus, it is called the IUT or intrauterine transfer. And these two methods, ZIFT and IUT, these are to be done and these are called the ET or embryo transfer. Now let's see that in vitro fertilization, first fertilization is done in the lab and then the embryo transfer is to be done. Now let's see the options. So embryo only up to 8 cell stage. Here we have to see that we have to transfer the embryo into fallopian tube. So first option given is embryo only up to 8 cell stage. Second is either zygote or early embryo up to 8 cell stage. Third is embryo of 32 cell stage. And D is zygote only. So I told you that in case of ZIFT, either the embryo up to 8 blastomere or the zygote, these both can be transferred into the fallopian tube. So either zygote or early embryo up to 8 cell stage is transferred into the fallopian tube, and this technique is called the ZIFT technique. While if the embryo, means this one is correct, but zygote can also be transferred, so it is discarded. But if the embryo of 32 cell stage is to be transferred, means at the blastocyst stage, then it is to be transferred into the uterus directly, and this technique is called IUT. While zygote only, so embryo can also be transferred, so this one is incorrect, this one is incorrect. So the only right option here is option B. So it is our right answer. I hope this one is clear. All the best.